What is up? It is February 1st, 2022, and some of you might have noticed, whoever watches my channel, that I already posted a video for today. Well, that was done late last night because I was attempting to exchange pieces of my schedule and try out different things at different times, but I found out that actually I think doing this eternal life study / neurology study in the morning is probably the best time for me to get it done because it doesn't interfere with the day. I typically do it before my work day starts, and I can not have to worry about it the rest of the day and just getting it kind of done with. Okay, not that I don't enjoy it. I enjoy it a ton, but I want to make sure that other priorities don't get in the way and I don't miss doing it. So anyway, so yesterday or today, I talked more about the synapse and some of the terms involved in talking about the synapse, like the synaptic cleft, the transmission acetylcholine, which is a neurotransmitter, the process by which acetylcholine is packaged in neurotransmitter vesicles, how those vesicles fuse to the membrane of the membrane of the synapse from inside to outside and release the acetylcholine into the synaptic space, the synaptic cleft, via calcium stimulation, because calcium channels open up within your synapse, which causes these vesicles, neurotransmitter containing vesicles, to release the vesicles outside of the cell, or outside of the synapse. And basically what happens when that acetylcholine binds to the postsynaptic cell, in the case that I was talking about, the muscle cell, and causes sodium channels to open up. And the acetylcholine itself is recycled by acetylcholine esterase once it's bound to these channels, and that makes sure that the acetylcholine is deactivated, because the acetylcholine being active causes the action potential of the postsynaptic cell to continue to fire, and it needs to be shut off essentially. And then those acetylcholine pieces are recycled. So to give a little bit more formality as to what's going on, and I might jump around a little bit because this is more complicated, but when we're talking about signal transduction, which is the general process, right, by what happens when hormones or stimuli from electrical impulses from neurons or any other signaling mechanism gets received by a downstream cell. And in this case, the downstream cell, in the case of the acetylcholine, the downstream cell was a muscle cell with receptors that we talked about. In general, it could be other cells too, other neurons or other muscle cells, but basically the signal itself is relayed ultimately in a synapse. Remember how the synapse works, right? The synapse has what we just talked about, a section where these chemicals are released, so electrical signals aren't directly going to the downstream neuron. The action potential releases hormones, which then chemically signal gateways, right? These chemically signal gateways are activated, and so the hormone, in the case of the acetylcholine, the hormone acetylcholine is a primary, or neurotransmitter acetylcholine is a primary messenger because it's sending the first part of the signal. But by the time the signal gets to the receptor on the postsynaptic cell, it's called the downstream function of that receptor, or downstream function of that initial signaling mechanism from the primary messenger is called the secondary messaging pathway. And so that's actually fairly involved because that involves a number of intermembrane proteins. And so one intermembrane protein complex channel that we talk about are one type is called Adrenergic receptors, right? I cannot pronounce that right now, but I'll get it eventually. So adrenergic receptors actually respond to a neurotransmitter called epinephrine, or adrenaline as most people know it, or norepinephrine. And so you have alpha and beta type adrenergic receptors. Regardless, what happens is, in that case, these secondary messengers essentially open, they go through the process of activating protein kinase, right? So that's the end step, but there's several steps in between, right? So what happens, this is a good example, general example. What happens, a ligand, which is the neurotransmitter, binds to this adrenergic receptor complex. It opens this up and allows a protein subunit. You can imagine there's a subunit attached to this adrenergic receptor called the G protein with alpha, beta, and gamma subunits. This messenger causes a conformational change in this receptor, which then converts the G protein GDP to GTP. That GTP then travels over to another intermembrane protein that's not exposed to external, the extracellular fluid, but which is contained within the exposed to the cytoplasm in the cell, called adenylate cyclase. This adenylate cyclase then, through interaction with ATP, then converts the ATP to a molecule called cyclic adenylate monophosphate, which then activates protein kinase. So protein kinase is part of the standard cellular response, right? So this is how you get the cell to do work, right? Because the protein kinase phosphorylates other proteins downstream and causes them to go through cellular processes. So basically, what you have this multi-step process. Just remember, messenger to G protein, and then G protein to GTP, and then GTP activating adenylate cyclase, which turns it into this molecule CAMP, which is cyclic adenylate monophosphate, and that activates protein kinase. So that, remember, here to here. And so that's basically what happens. Now, there's many, I'm going to jump around when talking about synaptic transmission and whatnot in cells. I'm cutting this video short today because I have a lot to do, but every neurotransmission pathway has different versions of this depending on what is the neurotransmitter. So once we get more into that, I'll get deeper down that rabbit hole and we can delve into the mystical, magical world of neurotransmitters. One thing I want to do is actually talk upstream about the presynaptic process and what happens there, because the neurotransmitter is not simply always converted, is not simply always put in a vesicle and just sent outwards. In the case of epinephrine, you have a pre-neurotransmission packaging process which converts tyrosine to dopamine. So we need to talk about that. I skipped a few things here and there, but also I'm learning about these things as we talk. So yeah. So that's it. I hope you have a good day. I will talk to you later. Bye-bye. Short video for today.